Okay. Okay. So this is chapter 37 in your textbook. And you know what? Yeah, I think it is the last qualitative disorders of platelets and vasculature. That's chapter 37. Yeah. The next thing we do is our review, and then our exam 4, and then our presentations, and then the final exam. So yeah, it is the last coac lecture. So we're getting closer to the end here. So that's good. Um, and okay, so everything is in chapter 37, qualitative disorders of the platelet. So this chapter is going to describe platelet and vascular disorders. Um, these are mostly platelet function disorders, and mostly what the patient is experiencing in this kind of disorders is going to be excessive bruising and also mucocutaneous bleeding. So petechiae, purpura, ecchymosis, and things like that. Some patients also have epistaxis and menorrhagia and hemarthrosis, which is bleeding into the muscles and the joints. But most times the platelet count for these types of disorders is normal. These can be congenital disorders as well as acquired disorders, but I'm only going to touch on like 2 or 3 of these only for this particular chapter. So in this page right here, I have a box from your textbook, chapter 37, that explains the qualitative abnormalities that are found in of platelet function that are found in some types of disorders. So these are mostly rare disorders. And again, the ones that I'll talk about are congenital, but there are some acquired disorders. So when we talk about platelet function, we're talking about the main functions, the major functions that platelets perform. So they adhere, they aggregate, they secrete substances. So those are the major functions that the platelet does. So these are disorders that have to do with those major aspects of the function of platelets. So we'll touch on a disorder of platelet aggregation, which is Glanzmann thrombocythemia. Disorders of platelet adhesion, such as Bernard-Soulier syndrome. And then we'll talk about some storage pool diseases. The book has pretty much covered all of these disorders right here. But again, we are not going to touch on all of them. We're just going to touch on 3. And then you're only responsible for learning these 3 for now, okay? We'll go back to these in other times when we talk about megakaryocytes and the function of platelets when we are in hematology. So for now, this is it. Okay, so excessive bruising and mucocutaneous bleeding. In a patient whose platelet count is normal, is indicative that there is some sort of qualitative disorder going on these patients. So again, you have a platelet count that's normal, but then there's some sort of bleeding issue going on, right? So then we're thinking more, well, the count is fine, but then there's some sort of functionality issue here that's happening with the patient. And so these actually didn't all get discovered until the 1960s when testing was, getting more specific over time. So a lot of these genetic issues were starting to be discovered, and these disorders were beginning to be understood because they weren't in the past, and so they were treated like general disorders. And now the mutations for most of these have been found, so they are attributed to these kinds of disorders, and so treatment can be applied to the patients that have these. Okay, so just a review here of the entire platelet and the components, the surface components and the intracellular components, so that you understand how intricate the platelet can be. And you already know this, I'm sure. But although it's a really, really tiny cell and it's anuclear, right? It doesn't have a nucleus. It has a lot of different functions, right? And platelets, again, serve are very, very important. That primary platelet plug that we need to form when there is vessel injury. And so when there is something that is not in place, or there's something that is deficient or abnormal, um, we are going to see the consequences. Okay, so when it's a genetic issue, then we are thinking about the way that this disorder has been inherited, right? So most of the time, the people that are the homozygous, that have inherited the homozygous form of this disorders are the ones that pretty much suffer with a full-blown disease, right? But the people that are heterozygous, it depends on the situation, but most of the times they have some sort of amount of the protein, the receptor, the signaling, but it's not at its highest level. So there could be some sort of consequences that they experience as patients. But let's go over all of these features that the platelet has. So you know platelets have, let's start all the way at the bottom. You know that they have alpha granules and they secrete substances, right? So there are diseases that have that are related to the alpha granules, like gray platelet syndrome and Quebec platelet syndrome, also alpha dense granule storage pool deficiency. These are disorders that have to do with the alpha granules in the platelets. There is a platelet type van Willebrand's disease in which there's spontaneous binding of van Willebrand's to glycoprotein 1b alpha. There is Bernard-Soulier syndrome, which is the lack of adhesion to van Willebrand's factor and abnormal response to thrombin as an agonist. There is Glanzmann's thrombasthenia, and Glanzmann's thrombasthenia, what you see is there is no aggregation of the platelets with the natural agonist. So you'd have to add a little bit more to see some sort of response. There is Hermansky Poldrak syndrome, Cherry-Akigachi, dense granule storage pool deficiencies, and these have to do with the dense bodies in the platelets. And there's also issues with signaling pathways, Scott syndrome, issues with the cytoskeleton, such as giant platelet syndrome and Wiskott-Aldrich syndrome, which we'll discuss a little later, and also issues with the enzymes of the platelets and the platelet cycle. So cyclooxygenase, thromboxane A2 synthase, and lipoxygenase. So there's so many things that can happen. Although remember, all these platelets are really tiny and they are anuclear. They have a huge function in our body, and there's so many parts to it that can lead to qualitative deficiencies, so deficiencies in the function of the platelets. So just a recap of platelet aggregation, because this is important when we talk about qualitative platelet disorders, because a lot of times these deficiencies don't allow the platelets to aggregate. So we have to perform aggregation studies, because that helps us diagnose and then determine the kind of dysfunction that is happening with the platelets. So you all remember that you can do two types of platelet aggregometry. You know that overall it is a very difficult test to perform. It's considered to be a high complexity test. It helps detect functional defects of platelets. It measures platelet aggregation in whole blood or in platelet-rich plasma, depending on the methodology that you're using. If you're using PRPs, your samples have to be kept at room temperature for 30 minutes after you centrifuge them before you test them, but must be tested within 4 hours of collection. Okay. For light transmittance aggregometry using platelet-rich plasma, it uses about 300,000 platelets per liter in the patient's plasma, and it measures an increase in percent transmittance as the platelets aggregate. That's light transmittance aggregometry with PRP. You can also use whole blood for aggregometry testing. When you use whole blood, you're using the impedance methodology. So you take a sample and you dilute that in saline, your whole blood sample, you dilute that in saline, And as the platelets form aggregates in the instrumentation, they collect on the two electrodes that are placed in the sample, and it changes the amount of current between the electrodes. So the change is directly proportional to the amount of aggregation that is occurring in the sample. All right, so optical aggregometry is the same thing as light transmittance aggregometry. Oops, sorry, I don't know what I just did. Oh my God, what did I just do? Hold on. Okay, there you go. All right, so again, light transmittance is the same thing as your optical aggregometry, correct? Okay, so for light transmittance aggregometry, we already know that there are certain instrumentation that it's available for light transmittance. You take platelet-rich plasma. There's a stir bar in the sample. You will incubate that sample for 5 minutes at 37°C. Then you will transfer to your testing cuvette, to your first cuvette. The instrument will direct focus light through the sample cuvette to a photodetector. And as the platelet-rich plasma is stirred, the recorder starts tracing, or the tracing stabilizes to generate a baseline near 0% light transmittance. And after a few seconds, once you add the agonist for this particular test, it'll start to trigger aggregation. Okay, when your sample is normal, you'll see that it goes through the different phases. There is a shape change and secretion of the platelets, and the percent transmittance that is occurring at the time of testing is monitored continuously, and it's also being recorded continuously. So if no platelets are aggregated, there will be no light transmittance because the platelets are still all over the sample. And if platelets are aggregated, then there will be increased light transmittance, right? So as more platelets aggregate, more light transmittance that will be occurring in here. So remember this, we start at the baseline with the resting platelet at a stable baseline at 0% transmittance. And then a shape change after the addition of the agonist will occur, okay? And then you get primary aggregation. Okay, the release or secretion of ADP and ATP from the platelet granules, and then a second wave of aggregation that forms the large platelet clumps, okay? That's the second wave of aggregation. The percent transmittance is, or the percent aggregation is measured by the amount of light through the test sample. So basically, this is a normal looking curve right here that you're looking that you can see in aggregometry. So what you're expecting to see are these 5 phases of platelet aggregometry if the sample is normal. Remember that the agonist has to be added. There are reagent agonists that will trigger aggregation, and they have to be added to our sample. Now here is an example of what a aggregation study would look like with a sample with using ADP as the agonist. A deficient aggregation would look like a plain line, and a normal aggregation in light of this agonist would look like it has the 2 main stages. So it's 5 phases in total, but you have the primary and then the secondary aggregation. It's something that we need to note. Now, this is for ADP. It could be slightly different in the phase of other agonists. So this just shows you sort of like a control or an example of what it would be like in the presence of collagen, arachidonic acid, epinephrine, and ristocene. And these are all agonists that are added to your aggregation studies. So the agonists that are chosen are really the ones we would have to look at the patient history and really try to hone down what we're looking for so that we can understand the type of aggregation with the agonist that we're going to be adding. Okay. And then this is just the example. This is like a review of what you've already had, but this is an example of whole blood aggregometry. And again, that's measured by impedance. And what you do is you take your whole blood sample, but you do have to dilute that in saline. And as the platelets aggregate, they collect on the electrodes placed in the sample, which change the amount of current between the two electrodes. And then the change is directly proportional to the aggregation in the sample. So the change is directly proportional. You could use either one of these, light transmittance or whole blood aggregometry. These are the two more common methods of platelet aggregometry that is being employed, but there are other methods of aggregometry. Okay, so the first condition that we're going to talk about is the Glanzmann thrombasthenia, GT, and this is a bleeding disorder, okay? This is a type of thrombasthenia in which there's a lot of hemorrhage, and the bleeding can be severe, and also it can be disabling for the patient. What's happening here is that this bleeding disorder is associated with abnormal in vitro clot retraction, but the patients that have this have a normal platelet count. So what's clot retraction? Clot retraction is the process by which the volume of a formed clot is reduced through the contraction of the intracellular acting myosin cytoskeleton of activated platelets incorporated in the clot. This is an inherited this disorder is inherited as autosomal recessive. Heterozygotes, so people that only inherit one copy of the bad gene are clinically normal. However, the homozygotes, those are the ones that have serious bleeding problems and complications in this particular with this particular condition. It usually appears or starts occurring in the neonatal period or in infancy, where people would see that excessive bleeding happens after circumcision. And that can also come with epistaxis and gingival bleeding in the infant. But people get petechiae, purpura, menorrhagia, also gastrointestinal bleeding, and hematuria, which is blood in the urine. So this is known as a type of condition that the severity of it will decrease with age. It will decrease with age. So it is suspected when this is happening to a very young infant or a young, a neonatal patient. So what's occurring is that there is deficiency or an abnormality of the platelet membrane glycoprotein 2B3A. So it's the whole complex that is missing. So it gets a little bit more difficult than this. There's more to it because this complex has subunits, but we don't really need to go into that. We just really need to understand the general information about this condition. So glycoprotein 2B3A is either abnormal or is deficient. This is a membrane receptor that is capable of a platelet membrane receptor that is capable of binding fibrinogen, von Willebrand's factor, fibronectin, and many other adhesive ligands. And failure to bind to all of these proteins and ligands results in a profound defect in the hemostatic plug formation. So there is no aggregation here. What's happening is that the glycoprotein 2B3A in the platelet is deficient or is abnormal. Again, homozygous individuals lack surface expressed glycoprotein 2B3A, and heterozygous individuals will have about 50 to 60% of the functional glycoprotein. So they still have it. That's why they could be clinically normal. This disorder, Glanzmann thrombasthenia, is a result of a genetic mutation, two in particular, the ITGA2B gene and the ITGB3 genes that are sitting on chromosome 17. These two genes code for glycoprotein 2B3. So mutations in these genes will lead to a deficiency in this glycoprotein membrane receptor of the platelets. Okay, so again, when you're looking at a patient that has this in the lab, what you'll see is that they have bleeding symptoms, especially the homozygotes. So they'll come in with bleeding abnormalities. Perhaps they're younger patients, but the platelet count is normal. Okay, so that's what leads you to think that there is some sort of abnormality with the function of the platelets. The morphology of this disorder will also be normal, meaning the platelet is tiny, it's still purple, it's still, it still has the same features as a normal platelet. So the count and the morphology are normal. When you perform platelet aggregometry, though, what you'll see is that it lacks the aggregation of platelets in response to all of the platelet activating agents. So no response with ADP, collagen, thrombin, and epinephrine. So because there's deficient glycoprotein 1B9, 2B3A. You have to stimulate this with very strong agonists such as thrombin to induce at least something in the aggregation phases. So you will induce with thrombin the release and secretion reaction, but that'll be in the absence of total aggregation. So there'll be some sort of reaction to it, but it's still not complete aggregation. Restocing and induced binding of van Willebrand's factor to platelets and the resulting platelet agglutination are normal in this particular condition. So restocing and induced binding of van Willebrand's to platelets and the resulting platelet agglutination are normal, okay? But we do lack aggregation to the activating agents, the agonists, the platelet activating agents, such as ADP, collagen, thrombin, and epinephrine. Okay, so this is a bleeding disorder. And again, it can be very, very debilitating for certain patients. The hemorrhage can be severe. It can be very disabling for these patients. And basically, all types of bleeding has been reported for patients that have GD. So all types of bleeding. So mucocutaneous bleeding, gingival bleeding, petechiae, bruising, all kinds, basically. So for the most part, the patient will have to be treated with normal platelet transfusion. So there is a way to separate the platelets and to just transfuse platelets to the patients because their memory receptors are not working and there's dysfunctional platelets perhaps. So they need these normal platelets in their circulation. So normal platelets will be a mode of treating these patients, but you may need repeated sessions for these patients because of defective platelets from the patient with this condition, and those defective platelets can interfere with the normal transfused, sorry for that typo, transfused platelets, okay? Another thing is when there is a lot of transfusions going on in the patient, you always have a risk of alloimmunization, especially with the platelets. So to avoid alloimmunizations with the human leukocyte antigen, it is important to use pre-storage leukocyte-reduced apheresis platelets, or you can also use HLA matched donor platelets as well. So HLA matched donor platelets may be a little bit more difficult, but using pre-storage leukocyte-reduced apheresis will really minimize the chances of alloimmunization if you are using repeated transfusions of platelets on this particular patient. Okay. At all times, patients that have this condition should avoid anticoagulants and antiplatelet agents such as aspirins and the non-steroidal anti-inflammatory drugs. There are a variety of treatments that have been used successfully to prevent the bleeding in combination of these platelet transfusions that we have to give these patients. But the way that they will approach this is by looking at the site of hemorrhage. Where is the patient bleeding from? Where is the major issue here for the patient, right? Because we said that all types of bleeding have been reported for these kinds of patients. So they will look at where, beside from giving them platelet transfusions, it's also where is it happening from, and this is how we can take it. We can take it from this point on. So the site determines the therapeutic approach. Hormonal therapy has with noriestrone acetate has been reported to have been used in patients to control menorrhagia, so heavy menstrual cycles. And so that has worked for those particular patients because heavy menstrual cycles can lead to, if they're very heavy and debilitating, they can lead to anemia in the patient. So that has to be treated when that occurs. And anti-fibrinolytic therapy can be used to control the gingival hemorrhage or excessive bleeding after a tooth extraction or any minor surgery. And other methods of therapy, such as recombinant factor 7 or R7A or Novo7 or Novo, have been used in surgical and non-surgical settings, especially in GT patients who are refractory to platelet transfusions with or without platelet antibodies. So refractory to platelet transfusions means that no matter how many units of platelets are being transfused to the patients don't seem to be able to get their platelets to function once again. So these conditions can happen because of certain antibodies that sometimes we create to the platelets, or just simply because the platelets they're not responding to the therapy or to the transfusions. And so for these cases, recombinant factor 7A has been reported for use in these patients, and it's proved useful for treating severe bleeding in patients with ISO antibodies to glycoprotein 2B3A and in patients undergoing invasive procedures. Okay, stop me at any time if you have any questions. The next condition is Bernard-Soulier syndrome, BSS. This is also called giant platelet syndrome. It is a very, very rare disorder of platelet adhesion. So before we were looking at platelet aggregation as one of the functions of the platelets, now we're looking at platelet adhesion. Platelet adhesion is very important. We need our platelets to adhere to the subendothelium so that they can participate in that primary platelet plug, right? So this condition also manifests in infancy or childhood with ecchymosis, epistaxis, and gingival bleeding. Hematorosis and expanding hematomas are rarely seen, so we really mostly see some superficial mucocutaneous bleeding here. It is inherited as an autosomal recessive disorder with the glycoprotein 1b95 complex missing from the platelet surface and or with abnormal function of this particular glycoprotein. There is an inability to bind to von Willebrand's factor and to adhere to exposed subendothelium. So this platelet membrane glycoprotein is so important, and you can tell because it just has this function of adhering to the subendothelium and to binding to von Willebrand's factor, right? And if we can't do that, there is going to be no primary plug, no adhering to that site of injury. And so this genetic disorder leads to this complex, this 1b95 complex missing from the platelet surface or having abnormal function. So again, there is inability to bind to von Willebrand's factor and to adhere to exposed subendothelium, and it results in bleeding for the patient. This condition actually resembles von Willebrand's disease. I don't know if you guys remember from the last lecture on Friday, we talked about the different types of von Willebrand's disease and what is happening. This is also an inherited disorder. Heterozygotes have 50% normal levels of glycoprotein 1b95 and near normal platelet function, but homozygotes, on the other hand, they see the clinical issue where they have enlarged platelets. They do end up having thrombocytopenia, which is decreased platelet counts, decreased platelet survival, all of which lead to severe bleeding disorders. The count for these particular patients can get decreased, so it can go from 40,000 all the way to a normal count to anywhere between 150,000 to 450,000. In the peripheral blood, what you'll see is large platelets that have a large number of cytoplasmic vacuoles and membrane complexes. And megakaryocytes in the bone marrow exhibit an irregular demarcation membrane system. So they look very large and irregular. So what do we see as far as aggregation studies go for Bernard-Soulier or giant platelet syndrome? With the aggregation pattern, you see that it is deficient in the presence of rhistocein because it lacks glycoprotein 1b95 complexes, and there is inability of these platelets to bind to von Willebrand's factor. Therefore, you see the clinical symptoms of bleeding. However, the difference here is that it will have a normal aggregation pattern with collagen, epinephrine, arachidonic acid, and ADP. Okay, so the issue is here with glycoprotein 1b95. There is no specific treatment for Bernard-Soulier syndrome, but it's been treated with, again, platelet transfusions and monitoring for the risk of autoantibodies, so using pre-storage leukoreduced platelets or apheresis platelets. Most recently, there's been the use of desmopressin acetate and recombinant factor 7 or NOVO7. And here is a picture of the giant platelets that you would see in a case of Bernard-Soulier syndrome. And these are your red blood cells right here, and this is a platelet. It looks like a cell. It's not supposed to look like this because platelets are really very tiny and they look like purple dots basically in the peripheral blood. So this would be a picture of high power field under the microscope. And when the platelets are the size of a red blood cells or bigger, they're called giant platelets, okay? But these platelets here have, they look like your typical platelets, but they're just giant in size. Okay, other conditions include secretion defects. So secretions, all our secretory substances that we see when platelets get activated, there's defects that have to do with those substances. They can be acquired defects. Some acquired defects are due to drugs or inherited defects. And these are defects in granules or in the ikonozoid pathway in which we experience the generation of thromboxane A2, which I explained to you guys earlier in this course. What happens in these conditions is that there's partial inhibition of aggregation, and there is an absence in the secondary wave with ADP when that's used as the agonist. Rhistocein aggregation with rhistocein will be normal, but with the other ones, there'll be various different patterns because there is partial or delayed response with the agonist. Okay, and then the next disorder is called the Wiskott-Aldrich syndrome. This is a rare X-linked disorder that is caused by a mutation in the gene that encodes for a 501 amino acid protein, and that protein is called the Wiskott-Aldrich syndrome protein, WASP protein. This is a protein that is found in hematopoietic stem cells, specifically lymphocytes, immature lymphocytes. So what happens here is that the T cell function is defective because of abnormal cytoskeleton reorganization, impaired migration, adhesion, and insufficient interaction with other cells. So this is all happening in these kinds of lymphocytes due to this mutation in the gene that encodes for this particular protein. The classic form of Wiskott-Aldrich syndrome was characterized by the susceptibility to infections that were associated with immune dysfunction with recurrent bacterial, viral, and fungal infection, microthrombocytopenia, and severe eczema. For these patients, thrombocytopenia is present at birth. But full expression of this condition occurs usually after 2 years of age. They lack the ability to make anti-polysaccharide antibodies, which leads to the propensity for pneumococcal sepsis. Patients also are at risk of developing autoimmune disorders, lymphoma, and other deficiencies or malignancies, which can lead to an early death. The thrombocytopenia that is experienced here in this particular condition occurs from something called ineffective thrombocytopoiesis. So ineffective thrombocytopoiesis is when, remember that the platelets are made in the bone marrow, right? So the bone marrow is the megakaryocytes are making these platelets, but they are ineffective in a way that they don't have the ability to release the mature platelets into the peripheral circulation. So basically, these megakaryocytes and these platelets are dying in the bone marrow before they can be released as mature forms, right? So a mature form would be the proplatelet processes dropping the platelets into the peripheral blood. So that is not happening because because of this gene, this genetic mutation, there is ineffective thrombocytopoiesis going on. And also there is increased platelet sequestration and increased platelet destruction. The bleeding can be moderate to severe. It's found primarily in males because it's an X-linked disorder. And the platelets are abnormal. They have decreased dense granules and they are small in size. So when you do platelet aggregation studies, you'll see that there is decreased aggregation with ADP, collagen, and epinephrine, as they lack the secondary wave of aggregation. But you will see that there is a normal response to thrombin in platelet aggregation studies. Because there is platelet sequestration here, one of the treatments for thrombocytopenia for this particular patient is going to be splenectomy, because especially when there is lots of platelet destruction and sequestration in this particular case. When the episodes are, when there are moderate to severe bleeding episodes, platelet transfusions will be needed, and bone marrow transplants have been attempted with some success. Okay, so that's Wiskott-Aldrich syndrome. And these were the qualitative platelet defects that we just talked about: Bernard-Soulier, Glassman thrombasthenia, secretion defects, and von Willebrand's disease. Von Willebrand's disease was actually something that we talked about last Friday. But what I want you to do is just look at this table here and try to make sense out of the table, understand what is happening and why the tests are a little bit different, right? So you have Bernard-Soulier syndrome, where there is a defect in glycoprotein 1b95. The platelet count is typically decreased. There is an abnormality with adhesion, so your PFA 100 will probably be increased. There is normal aggregation with ADP, collagen, and epinephrine. Your von Willebrand's ristosine cofactor von Willebrand activity assay is normal, but it is abnormal with RIPA, with ristosine-induced platelet aggregation studies, because of the abnormality that is occurring in glycoprotein 1b95. Other things that you see in Bernard-Soulier are the giant platelets in the peripheral blood. With Glassman's thrombasthenia, you have the abnormality or deficiency of glycoprotein 2b3a. The platelet count tends to be normal. The PFA 100 will be increased because it is an aggregation problem. There is no aggregation with ADP, collagen, and epinephrine in aggregation studies. Your von Willebrand factor activity assay is normal, and your RIPA is normal, and there is no clot retraction. That's the abnormality that is occurring. So this is just a table that helps you differentiate between the two, because Bernard-Soulier and von Willebrand's disease can be very similar to one another, but they have different testing. For example, if you did rhistocene cofactor assay or van Willebrand's factor activity assay, it would be decreased for van Willebrand's disease as opposed to as opposed to someone with Bernard-Soulier syndrome. So it's good to be able to determine the issue here, and to not just memorize this table, but to understand what the test is doing and why my patient with this disorder is positive, negative, decrease, increase, or whatever the case may be for this particular, for those particular tests. Remember that rhistocene-induced platelet aggregation is a test that we use to determine the presence and the integrity of platelet glycoprotein 1b95 and van Willebrand's factor. Okay, so you're looking at the interaction of both of those when you're doing RIPA. And you usually perform platelet aggregometry with that using platelet-rich plasma. All right. All right, so do you guys have any questions? This is a short lecture, it's not very long. I do have an activity for you guys to do. It's already posted in the course, but we don't really look at that until Friday. And that's about it. Any questions for me?